I had a wonderful email from a former student in my lab who is now studying for his MD PhD. But at the time he was working in our research lab here at UC Berkeley, and he reminded me of a wonderful day when I was, which in retrospect is wonderful. At the time, it was just business as usual, right? I was in the lab with a lab member of mine, Martin Yinek, who was the person who was doing work on a protein called Cas9, part of the CRISPR system. We call it CRISPR-Cas9. And we were trying to figure out how it worked. It was pretty clearly an RNA-guided enzyme that could find and cut DNA. And the question was, how does it do that? Does it do that? And if it does, how does it work? And he was doing experiments to answer that question. And he eventually figured out that this system could be programmed literally with different segments of RNA that have the same sequence of chemical letters that match those letters in DNA. That's what allows it to be programmed to find and cut DNA at a particular place in a cell. And this student of mine, whose name is Prashant Bhatt. So Prashant reminded me of a day when he came in the lab to do his experiments, and he saw me and Martin Yinek leaning over a light box in our lab, which was a way of visualizing the data from one of Martin's experiments, and which we were just looking at each other and high-fiving, and we were just so excited. And why? It was because that was the first experiment where Martin had showed how the system could be programmed and also how we could simplify it compared to what nature had done. We turned it into a two-component system, just one type of RNA and one protein that were necessary for this RNA-guided DNA cutting reaction to work, this chemistry to happen. And I think for us, that was really that precious email from Prashant Bhatt reminded me of that, one of those kind of moments that we all live for as scientists, where you make a discovery that you realize is profound in a way. It's much bigger than the experiment began. It's something that could have big implications because of the way it works. Well, whether we really did that is probably up for debate. And the reason I say that is because knowing Martin Yinek, he's a very subdued individual. He's very scholarly, and he would always try to, I would get very excited about things, and he would always say, "No, Jennifer, no, we have to wait. And I've got to do another control, and I've got to repeat that experiment. Before we get excited, let's make sure." CRISPR is really it's a fabulous tool for doing surgery on DNA. And it's basically a way to alter the code of life. It's a way to change the DNA sequence in cells so precisely that we can now alter a single letter in the code of a human cell. Human cell has 6 billion individual letters, 3 billion base pairs of those letters. We can change a single one with this technology and do other things as well. So I think that this has given scientists a way to rewrite DNA that provides opportunities both for fundamental research to understand what the DNA code is telling us about the way life is, as well as to, in the future, do things that will probably correct disease-causing mutations, create plants that are better adapted to their environments, and many other things. Well, there's many, but I think the ones that are probably closest along include curing sickle cell anemia, which is a disease that involves a single letter that needs to be corrected in a single gene of a human cell, a human red blood cell. That's something that has already been cured in cells growing in laboratories and also in animal models. And we're now at a point where several groups are about to begin clinical trials in people. BRCA1, BRCA2, these are mutations well known to correlate with a high risk for breast and ovarian cancers in women. Right now, it would be difficult to use gene editing to correct that mutation, not because we can't do it in individual cells, but because it would be hard to correct it in every cell of the body. So I think that's frankly one of the current challenges with the technology, is just delivering it into cells and tissues. And the reason that sickle cell anemia is one of the first targets clinically is because cells can be edited outside the body and then replaced, and that can be done in a way where those cells then take over the blood supply in the person and replace the cells that are defective with cells that are corrected. One of the amazing things about this technology is that it's really opened the door to experiments that scientists are doing in many different kinds of organisms to understand how they work and why they are the way they are. What in their DNA makes wheat susceptible to certain kinds of funguses and or fungi. What is it in people that makes us have certain kinds of properties in our bodies? What in our DNA creates certain traits? I think that is something that maybe most people who are outside of science don't realize, but there's been an enormous uptick in the pace of scientific research that's driven by the power of this technology to allow scientists to ask questions in their laboratories that could never be asked in the past. I think many of us will by then, because I think we'll all be eating food and using crops and plants that will be genome edited. I think that we will be benefiting from, medical breakthroughs that will have happened due to genome editing, whether it's an actual therapy or not. We'll certainly benefit from the biomedical knowledge that's coming from using this technology to understand the cause of disease, for example, and really kind of start to get at this whole opportunity of personalized medicine. It really comes down to every one of us is a little bit different based on our DNA, and understanding that at a molecular level, I think is coming. And then I think also, we didn't talk about this, but benefiting from other applications of CRISPR, whether it's controlling the spread of mosquito-borne disease, whether it's making chemicals in research laboratories or in commercial settings that are made using bacteria or fungi instead of polluting chemical processes. I think that's also coming with CRISPR, sort of synthetic biology is what we call it. So I think within 10 years, we'll probably all of us will have many things that touch our lives that will come from CRISPR. Well, that question to me really brings up a couple of different but important points. One is that something like depression is not the result of a single gene or even a handful of genes. It's almost certainly the result of hundreds or thousands of genes, as well as the environment that a person has experienced. So it's going to be very complicated to try to, even if we wanted to fix it with genome editing, at least the way the technology is right now. And the other thing is that the other point about your question that's so interesting is that it sort of gets to this question of what makes us human, what makes us unique as individuals. What do we value about the diversity of our life here on Earth, and I think that for many people, the diversity of life is one of the things that makes it so rich and so wonderful. So we wouldn't want to have a way to make us all the same. It gets into the whole question of eugenics and sort of access, who decides, who regulates this, who makes those decisions, who pays for it, who should be allowed to do this, all of these kinds of questions. And again, I want to emphasize that the technology today doesn't allow that yet, but it's close enough that we know we need to be considering those questions. I would say I didn't get pushback, but it definitely we heard a lot of different opinions about the technology, as you would want, I think, in a conversation. I'm thinking in particular about meeting that was convened I convened with my colleagues in January of 2015. Small meeting up in the Napa Valley. We just had some clinicians, some fundamental researchers. We also had scientists who had been involved in the discussions of ethics of molecular cloning back in the 1970s were in attendance. And it was really a meeting to start grappling with, we've got this powerful technology of genome editing. It's moving incredibly rapidly in the scientific arena. And yet none of our government regulators at that time or many people outside of science knew anything about it. And so it was this weird kind of feeling of sitting on something so powerful and moving so fast and could affect many, many people. And yet most of them weren't even aware of it yet. Well, there was a wonderful comment that was made by a colleague of mine up there. So, I remember that, we were sitting around a table about the size of the table in this room and, debating in particular about this issue of human embryo editing. Should people do that? Is that something that we should be trying to work towards or work against, or how do we think about that? And a lot of people around the table were sort of talking about all the dangers of doing it and how risky it could be, et cetera, et cetera. And then at one point, a colleague of mine leaned across the table and said, wait a minute, at some point, this technology may be accurate enough that we'll all realize that it would be unethical not to use it that way, to correct the disease-causing mutation for cystic fibrosis or Huntington's disease, things like that. Yep. And it made everybody stop and think for a minute and just say, wow, we could turn this whole question on its head. Well, it might surprise you maybe, but the thing I feel most worried about today is really just I worry about the pace of the technology outpacing our collective ability to understand the implications of what it does. And but what I mean by that is that I think that for scientists to race ahead to do things that we can do, but to coin a phrase from that wonderful line in in Jurassic Park by Jeff Goldblum, love it. We can do it doesn't mean we should do it. And I think it's really that it's wanting to make sure that scientists are thinking about the implications of their work, especially with respect to genome editing, prior to just rushing forward. It's a complicated issue because science is global, of course, and people are working, in every part of the world now using CRISPR-Cas9 technology as well as, many other technologies. And how do we create a regulatory framework that people will honor in these different jurisdictions? How do we create even a community of scientists where there's a kind of a code of ethics that everybody would buy into? I think it has to begin with, in my view, it really does have to begin with kind of a grassroots effort. It has to begin with scientists coming together and discussing these topics. And I've been involved in convening meetings around this. And fortunately, I think that idea has really spread in the genome editing field to include now people, not just scientists, but also people that are stakeholders, whether they be, patients with genetic diseases or clinicians that hope to use this in the future, or agricultural scientists who want to use it in plants and other types of applications. So we are seeing a pretty broad cohort of people that are starting to attend these meetings and are interested in the topic. But I think the real challenge ahead is to figure out how to put all of that effort and energy into a cohesive document or consensus kind of opinion piece that people could buy into. Oh, certainly they don't agree on that. Not at all. I think for many people, the opportunity to correct a disease-causing mutation at the time of conception, rather than having to wait and do it much later, is an attractive idea. The difficulty is in thinking about all of the other implications of that kind of of genome editing, as well as the technical challenges with it. When I was in graduate school, I worked with a wonderful scientist, Jack Szostak, and that's how I felt about him. I felt like the thing that he had so wonderfully, and it was hard to, it was kind of one of those je ne sais quoi, hard to put your finger on it sorts of traits, is that he had that quality. I felt he knew the right experiments to do. He knew the right questions to ask. He didn't know the answers, because that's why we do science, but he knew the right ones to ask. And I always thought to myself, gosh, if I could bottle that up somehow and figure out what that is. And I never imagined that people would see me that way, but I know I really consciously tried to understand that about the people that I've trained with, because I think I have benefited from mentors who were able to do that very, very beautifully. I've always been driven by a fascination with evolution. Why is life the way it is now? And of course, we're seeing life on our planet at a snapshot in time. It was different in the past, and it'll be different in the future. And it's all strung together by DNA, right? It's the code of life is kind of evolving over time. And I've always been very, very interested in that process. So I feel like if there's a thread to my research over the years, that's kind of always underlying what we do, is thinking about evolution and how it works. I think that I've always sort of had a knack for experimental science. It's a feeling that I have about both the questions that I want to try to answer that I think are important, as well as the way to go about doing that. And I once heard my postdoc advisor, Tom Cech, another very wonderful, celebrated scientist, Nobel laureate, he once described me as somebody who had a map in her head of experiments that were to be done and could sort of see a whole process. And it was somehow when he said that about me that I kind of realized, gosh, I think he's right. That is how my mind works, and I hadn't really thought about it before. But I think that is what I would say about me, is that I always have had kind of a talent there of thinking about how I'm going to do experiments, and I still have it. When I meet with people in my lab, I find that even though sadly I'm not in my lab anymore doing actual experiments, I'm still good at thinking about how to do those experiments, which ones are important to do, and how to set them up so that we get data that are going to tell us something meaningful. It's not really visual. It's not something I could draw on a piece of paper. It's more a process in a way, right? It's it's almost like a flow chart, right? It's sort of thinking, okay, well, if we do, if we want to figure this out, we could do this, and then this, and then and then depending on how that works out, we can do this or this, that kind of thing, right? It's almost like a flow chart, and it's sort of, there's a logic there, right? And then I'll think, okay, and if we want to do that experiment, I know we need these controls so that we can interpret the data, and and then I'll start thinking about how to logistically, how to get all the pieces to, this person would be good at doing this one thing, this other person over here would be great to help us out with this other thing, that person over there would be a great collaborator because they know how to do something that we don't know how to do, that kind of thing, right? So I do love kind of team building and thinking about, like, if you want to answer a question in the lab, how do you do it, both which experiments to do, and also how do I bring the right people together to do those experiments? I love doing that. It's fun. I loved where I grew up. It was such a magical place in many ways. It was sort of a kid's dream. We had the freedom to run around in the afternoons, and we were definitely checking out our sugarcane fields, snacking on occasional cane stalks, but also exploring guava trees and running around in the huge tall California grass that grew back of our house. It was just a wonderful experience of the native world. Sleeping grass, yeah, that would sit there, and when you stepped on it, poke you, but then it would close up. Leaves would close up. Seemed like some kind of interesting chemistry going on there. I wanted to figure that one out. I felt like one of the neighborhood kids, and I just loved. I played kickball. I like to play kickball out in the street. I had a horse. I used to ride horseback and ride in 4-H, little rodeo events and things like that. So I just, I had a pretty active, very outdoor kind of life. I wondered why sleeping grass, closes when you touch it. What's going on? What's the chemistry there? I wondered about, I would walk on lava rock and I would see all these colors and shapes in the rock, and I would wonder why. What's there? There must and why did lava in one part of the island look so different from lava on other parts of the island? There must be different chemistry going on there. I was always thinking about the chemistry under it. Wanted to know about that. um, and then just about the plant life. I mean, there were so many fascinating plants in growing in Hawaii that I didn't see growing anywhere else, and I always wondered, why is that? So things like that. Well, my dad was his specialty was Henry David Thoreau, and so we had multiple copies of Walden around our house, and people reading Walden at various dinner tables. That was something that happened quite regularly. And my dad also loved reading all kinds of things outside of his specialty, including about science. And so one of the really cool things for me growing up was that without even really thinking about it, I got exposed to some really interesting science writers, people like Lewis Thomas and James Watson, the double helix, his book about the discovery of the DNA structure, and books written by medical doctors who were sleuthing the cause of disease in patients. So there were quite a range of things that my dad read or gave to me to read and that we discussed in our house that I think really gave me a sense of what it was like to be a scientist. Well, hindsight is always 2020. People now say, "Well, weren't you struck by Rosalind Franklin's role and how she was maybe treated by these scientists?" I didn't think about that at the time. Isn't that interesting? I was so captivated by the story of discovering the structure of DNA. I was so amazed by the possibility that you could, as a scientist, ask a question that detailed and figure out the answer. It seemed amazing. And I guess for me, that was the first time that I had a sense of science as a process. You know what I mean? As a a growing living thing that was created by people rather than, when you're in grade school, you have science textbooks that, at least in my day, were pretty cut and dried, and science was seemed kind of boring. It seemed like memorizing a lot of facts, and it didn't seem all that interesting to me. But when I read the double helix, that was the first time I had the sense of being a sleuth. You're on a trail, you're hunting something down, you're trying to figure something out, and you've got just bits and pieces of data, and you've got to try to put a story together. That was amazing to me. In high school, I had in 10th grade had a fabulous class, chemistry class. Miss Wong was our teacher, and she was very good at helping us kids understand the process of doing science. That science is experiments. You ask a question and then you figure out how to answer it in the laboratory. And so for me, I became very interested in chemistry, but I knew I liked the living natural world. And so those were just kind of nebulous interests of mine at the time. And then when I heard this lecture by a biochemist talking about her research into why cells become cancerous, it suddenly gelled for me. I realized that's what I want to do. I want to figure out the chemistry of living things. I want to understand the molecules that go awry in a cell that becomes a cancer cell. How does that work? I certainly didn't know any female scientists at that time, except, the famous ones, Curie, but I didn't know anybody anybody living that was a female scientist. I just didn't know them. And so it seemed really exotic to me to, first of all, see this lecture by a woman scientist, and secondly, to think to myself, oh, I wonder if I could do that. I wonder if I could be a scientist. And I still remember thinking, had this little chill of thinking that would be really, weird and wild to become a scientist. I wonder if I could really do that. Well, I think there's been a lot of advances for women certainly in my area in the biological sciences over the last few decades. And so I feel very fortunate that right now in my department at the University of California, Berkeley, for example, we have quite a good number of women at all stages of their career, from very senior tenured women to women who are just starting their research. But that being said, I find that we still see that in the more senior levels of academic science, as well as all across the business world, there definitely is a lack of women that you see. And I do experience things like, especially business meetings or higher level administrative meetings, where I find that there aren't very many women in the room. Occasionally, I'm the only one. There's been a lot written about that. I think there are some trends that I see that are different in different in the genders, between the genders, but generally I sort of again, I don't that's not really how I think about things. I tend to just think of myself primarily when I'm in a professional setting as in the context of my work and what I know about and what I can contribute to. I think that it is true, or certainly I've seen in my experience, that I think women tend to not trust themselves as much as maybe they should, not to put themselves forward as much as maybe they should. And so I always encourage all students in my lab, but especially women, if I see them maybe doing that, to tell them, yes, you are good enough to apply for that job. Yes, you should apply for that fellowship. Yes, please do apply to attend that graduate program. You are good enough to do it. And just to have self-confidence, trust themselves, and to try things, take a risk. I think sometimes women talk themselves out of things when they don't need to. It's genetics in action. It's hard to say, right? I mean, yeah, we're all really different in many ways. Not in every way. You can tell we're from the same family in certain ways, but I think that we all have really different interests, and that's something that's made our family fun. And I think I saw that in my parents too. Like I described my father, being a literature professor who was nonetheless very interested in science, and I could see in a different had he a different lifetime or there was an alternate universe or something, he could have easily been a scientist, I think. I like gardening. I love playing with plants. I'm a big tomato fan. I grow tomatoes at home. I grow flowers, herbs. So I do a fair amount of that. And also I love, I'm very much an outdoors enthusiast. So I do a fair amount of hiking, biking, running, things like that. I run 10K races occasionally with my son. That's fun. 100%. 100%. I have found that I tend to have my best ideas and creative moments when I'm not in the lab. When I'm doing something else, I'm thinking about something else seemingly, and suddenly I'll think, wait a minute, this would be a really cool thing to do in the lab. And I also find that a lot of times I'll, especially if I've been relaxing a little bit, or just, not focusing on work for some period of time, like either a little vacation, or even just taking a day off of work, or spending time on the weekends going for a hike, I'll often find that it's those moments that will stimulate a creative new idea. I'll wake up in the morning thinking about our science, and I'll suddenly think, oh, wait, there's something really interesting here. We should pursue that. I guess if we go back to the kind of early days for us with CRISPR, even before the molecules that we worked on that are now being used for genome editing, I was actually studying with a couple of my lab members how bacteria fight viral infection using a system known as CRISPR. And in those days, we were studying some of the proteins that are responsible for cutting up the pieces of RNA, which are the chemical labels that these proteins use to find other molecules in bacteria. And it was really, I had gone on a small hike, with a couple of my lab members up in, I think we were in Muir Woods, which is for those folks that know Northern California, you probably know Muir Woods, but if you don't, it's a beautiful, redwood forest that, was established many decades ago, that really has preserved the beauty of redwood trees here in the Northern California area. So anyhow, we were up there hiking, and in the course of that hike, we were just kind of chit-chatting about various things, not necessarily science. But when I got back from that hike, I was talking with one of the students in the lab, Rachel Harwitz, who is now a very successful CEO of a company that we founded together. But in that conversation, we talked about some of the work that we were doing in the lab. And I think for me, that was maybe one of the first moments where I started thinking that the work we were doing could be useful as a technology. Now, I wasn't necessarily thinking about genome editing at that moment, but I was thinking about ways that we could use CRISPR proteins to find pieces of viral DNA and RNA, and how we might be able to use that as a research tool. So for me, that was in a way the beginning of really thinking about these CRISPR systems, both as a curiosity-driven research project, but also as a potential source of new technology. I kind of get into a zone, I have a desk in my kitchen at home, and that's often where I'll sit, and I can look out at my garden, and that's always a source of inspiration at some level. But I'll often sit there when I'm working or thinking or reading. A lot of times I'm reading and I'm taking notes, and I'm kind of puzzling something out. Sometimes it's just I'm out weeding my tomato plants, and like I said, suddenly something will come to me, and I'll think, ooh. And then the other thing is that I found that I have to keep a notebook by the side of my bed, because it's not uncommon that I'll wake up at some hour of the day or night, early day or late night, and have an idea and think, ooh, that's interesting. I want to pursue that. And I'll just jot it down. And I'm usually scribbling in the dark. It's like I can't see. I'm just trying to scribble enough that I can read it in the morning and remember the idea. There's always juggling going on with everything, even at work. In my younger years, I was, a real lab rat. I just, I worked a lot, and, because I wanted to, I just loved it. I loved being in the lab. I loved the feeling of discovery, and, but I also had, I'm a realist too. And, students that might be watching this might be thinking, but science can be so frustrating. And I hear you, it can be, and so I think for me, it was critical that I had other things to, distract me from my frustrations at times at work. I always loved gardening, for example. I've always had a little garden growing wherever I lived. I've always done a lot of hiking and biking. And, when I lived in Colorado, I did rollerblading, and, just getting outdoors and, getting away from things that way. But nowadays, I have to balance my love of work and research in the lab with my son's needs, the fact that I'm, I have a husband at home and a family to, spend time with. And I also am responsible for my mother up until recently. And so, just things like that. So, I think it's always about balance. It's absolutely possible. And I have many friends who are also women, highly successful, have families, and, they figure it out. I think it helps to have a really supportive partner. That's what I've found. It's incredibly helpful. My husband is the most supportive partner on the planet. He's a great all around, parent, confidant, fellow scientist, colleague, romantic partner, all bundled into one. And so I think for us, we've always viewed our relationship as, we have our private life and our family and our son. And we've also have our science together, and we enjoy both. And we kind of wear different hats even with each other. And also my husband is very understanding about especially now, my crazy schedule, my travel. I travel a lot. And so he's the one, more than me, frankly, who is going to the high school meetings with my son and going to robotics competitions with our son, helping to coach their team, just all of the things that, you really need a parent to do. I haven't really done that. Maybe I should. We do read a lot in our house, and I brought a book home last night, and I had it sitting on the kitchen table. And my son saw it, and he said, "Oh, what's that, Mom? What's that book?" And it's a novel written by it's a novel, but it's written by a young man who, not so young anymore, more like my age, who is Vietnamese, and it's kind of a novel about the Vietnam War, and thought it looked really interesting. So we read all sorts of things in the house. My son actually loves math, and he's a bit of a math whiz. So the kinds of books that he tends to read for fun are actually, fun books about mathematics. And so those are ones that he turns me on to. I think that skipped a generation. My mother played the piano. I tried and kind of it never gelled for me. And now my son is a great piano player and also plays viola. And I asked him recently, if you somehow didn't pursue science, what else would you do? And he said, "Oh, I'd be a musician." Not really. It's kind of a science fiction-y kind of fear. Yeah, I think for many reasons, that's not something that we have to worry about right now or even in the near future. I do think that, making changes to the human germline is coming. The germline that means making changes to DNA that become inheritable, right? So they change the DNA not just in an individual, but in their children and all of their progeny going forward, all their children's children. So becomes an inherited trait. That's a profound thing, and there's no doubt that this is coming. This was it was announced at a meeting in Hong Kong last November by a scientist who said that he had actually made changes to the germline of 2 baby girls who were born. And I think that announcement catalyzed a lot of conversations, and I think that's really motivating people to move with a bit more speed and focus in terms of understanding the technology today, where it's going, and how we ensure responsible use. Yeah, it wouldn't be possible to do it for a couple of reasons. One is that federal funding is not available, cannot be used for any kind of human embryo work, not just genome editing, any kind of human embryo research. And the other is that the Food and Drug Administration that reviews any kind of clinical therapeutics or procedures is not allowed to even review a procedure that would affect human embryos, as we're talking about. We could certainly compare it to the early days of I would call it modern molecular biology, molecular cloning. This was back in the 1970s when scientists first began making copies of particular pieces of DNA, being able to clone the insulin gene, for example, to make insulin for people that are diabetic, and do that in an industrial setting. That was also revolutionary. It was sort of the birth of companies like Genentech, and now many other biotechnology companies that are creating the therapies that many people are benefiting from today. So it's sort of that. I would say we're sort of at another kind of revolution of that nature, where it's now possible to do things that just a few short years ago scientists didn't have access to. Partly that's probably for many reasons, but partly it's technical, quite honestly, right? I think cloning people turns out to be very hard. Even cloning more sheep like Dolly is not a trivial thing to do. It's not something that very many people have access to the tools to do or the knowledge to do. And that's where genome editing is a bit different. It's a tool that is widely available. scientists don't need to be specialists particularly to be able to use it. And that's one of the wonders of it. It's a great democratizing tool that has given scientists around the globe the power to alter the code of life in different cellular settings. But it also means that it's much harder to control it. What it does is it provides protection for the use of that idea or technology so that companies, if they want to commercialize that technology, they have some protection to do so and to create things and products and charge money for them without worrying about competitors immediately stealing their idea, right? That's sort of the whole goal of the patenting process, is to give opportunities for companies and for investors in those companies to be able to trust that, if they invest in a company, they can develop something without immediately having competitors able to capture all of that work and make money on it separately from them. And so what's happening right now with the CRISPR-Cas9 patent situation is that recently the University of California has had a whole series of our patents issuing. And we have quite a large number that are about to be issued. And so that's creating a situation where, we now have patents at the University of California, as well as patents at MIT Broad Institute, and frankly, many, many other institutions. And so the US Patent Office is going to have to, again, sort out, what's the primacy of those patents. And we expect that there will be another interference coming that will allow the patent office to ask that question in a legal sense. Interference means that you have two patents that make the same claims on some kind of technologies or claims that are overlapping. And so then there has to be a resolution of that. If that were to not happen for some reason, then you have what we have today, which is that the University of California has issued patents, MIT Broad Institute has issued patents, and there's some overlap in the claims of those patents. And so anybody wanting to use this technology commercially needs to have a license to both parties' patents. And so, it's not ideal. If you look at the market capitalization values of companies that have already are already traded in the public markets, they're in the billions of dollars and collectively, and there are many, many more companies that are using the technology, as well as companies that are getting started that will, build value over time. I think there are different scenarios for how it could play out. It's not clear yet. I think if this interference is declared, then that will provide a legal platform for figuring out really who invented this technology and who should have the primary patents to it. And if that doesn't happen, then it'll be sorted out in, probably with lawsuits in the courts eventually. Is the patent fight nastier than I thought? Yes. I mean, I think that's been one of the worst parts of this whole experience for me, honestly. I couldn't have prepared for it. And I've tried to view it as part of my education. It's a part of and sometimes education is painful. Sometimes the way you learn things in life is through pain. And this is one of them for me. It just is. But I try to view it as part of my education. It's part about learning about people and their behavior, learning about the patenting process itself and why do we do it and how does it work. And in many ways, I feel it's sort of broken, but this is the system that we have, and we have to work with it. And there also are occasional glimmers of light. I mean, I guess I was incredibly happy that my university has stuck up for me. They have. They could have let me just drift away. I mean, they could have just turned their back, and they didn't. And that meant a lot to me. Well, I would find a way to protect ideas in a way that would enable them more. And I guess I'm particularly referring to technologies like CRISPR that are frankly, so enabling to so many people that it's actually not a good idea to squirrel them away. You really want to get them into as many people's hands as possible and let them take over the creative process with that tool. Use the tool, whether it's a hammer or a gene editing molecule, use it to do creative things, build the house or build the cell, and make those products, and to do, to have a system that inhibits that somehow with a foundational technology like that is, it's not a good idea. How would I do it differently? It's very hard to say, right? It's, how can I come up with a better system? I ask myself that occasionally, and I don't have a good answer. But I do think that we do need to collectively come up with a system that will make technologies like this, and there are others, of course, and there will be ones in the future, make those widely available so that they can be advanced as quickly as possible towards curing disease and doing all sorts of other things that are beneficial to people. Oh, we always have. I mean, in fact, the foundational work that we did was done with Emmanuel Charpentier, right? And the publication of the first publication about Cas9 and showing how it could be used as a genome editing technology in 2012 was published with her and her research lab. So that collaboration was foundational and fundamental, I would say, to developing this technology. But you're right, we've also worked with many others in the field, and men and women. I love that about science. I love working with other groups. It makes it fun. Oh, it's incredible. I mean, it's such a privilege to work with people like that, minds like that, and that's something I just love about science. Thinking about big questions and trying to break them down into something you could eventually actually test in a laboratory. My graduate advisor's question was, "What's the origin of life?" Kind of doesn't get bigger than that, and yet he figured out how to do experiments that get at that question. Incredible. It's very exciting. It's a wonderful, wonderful job, a wonderful opportunity that I've had. I could never have imagined, that my career would take me to where I am today. Honestly, I really never could have imagined. I could not have planned for it. But here I am, and I feel so fortunate that I'm at one of the world's best universities. I'm at a public university. We have people coming from all over the world, all kinds of backgrounds that come to Berkeley because they love knowledge. They want to learn. They want to study. They want to do science. They want to understand the world better. And it's a privilege to work with them. Well, I'm most excited about two things. One is continuing to understand the fundamental basis for CRISPR-Cas technology and how these molecules work. That's what probably 2/3 of the people working in my research laboratory are doing, is mechanistic, dissection of these systems, trying to understand how these molecules do what they do. How do they find and cut particular pieces of DNA or RNA in cells? How does that work? And the other thing that we're doing is trying to figure out where this is all going in terms of how do we deliver these molecules into cells or tissues in a patient, for example, or even in a plant. And how do we ensure that when those molecules get into cells or tissues, they make the right kinds of DNA changes and not anything else? I had to pinch myself. it's a it's a wild thing. I have no idea if that's really true, but I think it was an acknowledgement that the technology of genome editing is profound, and I think it will have a very broad impact across the planet going forward, and that this is really an important moment not only for the science and the technology, but also for the responsibility of it. Thinking about how we use it ethically. I worked at it and I'm still working at it. It's important to me. I think that, it's very, very important for people to understand science and hopefully capture a little bit of the feeling of excitement that comes out of, the research that happens in laboratories and how it affects our lives going forward. I think that's critical. Well, people always say, if you want to make money, go do something else. Don't do academic science. That's not totally true. I mean, I've been fine. I mean, I even when I was a student, I was always able to save a little bit of money, put a little bit away. But I've always lived, pretty modestly, I guess. I have to ask myself, like, why do I do what I do? Am I doing it to get rich? Am I doing it to be famous? And the answer is no, I'm not. I mean, I'm doing it to make discoveries about the natural world. I'm doing it to satisfy my curiosity. If I'm ever doing it for any other reason, I should get out, I think. Well, I guess the best thing about them for me is that it's a way of saying to students and to kind of our society in general that we value science and that we value scientists. I think that that's one of the things that I've been sad about over my career so far is that, I've seen a kind of a growing distrust of science and scientists, and that makes me sad. And I think that for me, these prizes are a way of acknowledging the value that all of us, get from science, the importance of making fundamental discoveries, and of every now and then, for as we just discussed, I think that for academic scientists, it's a challenging job. We work very hard. We're not paid, like the CEOs of companies by any stretch. And and I think you have to do this job because you love it. You love the science and you love the students. And every now and then, I think it's nice that prizes are there to sort of say, hey, let's step out of the lab for a minute and let's just celebrate the fundamental joy of discovery and the power of science to advance our society. Well, what's next? Gosh, I mean, I think what I love about science is I don't know what's next. I think in the broadest sense, what will I be working on 5 years from now? I literally don't really know. I do think that we kind of collectively are at a really interesting moment in biological research where there's an integration of different technologies, including genome editing, but other kinds of things too, including computation, artificial intelligence. There's lots of interest in that kind of intersection, and I think there's a really exciting opportunity to steer that ship and kind of bring those different kinds of technologies to bear on questions that I think are interesting. And I find that what are those questions? Well, I'm certainly continually fascinated by what we, meaning me and my colleagues and collaborators, are finding about the way that microbial life populates the planet, all of the different kinds of genetic pathways that those microbes have. We know there will be new discoveries and technologies that will come from those microbes. I think that's one of the things that we're most excited about focusing on in the lab right now. And the other thing is really trying to build the ways that CRISPR tools will be useful both in clinical medicine and in plants. And that really involves ensuring that these molecules can be delivered into cells efficiently. And when they get there, they make the right edit. Well, I guess one of the things that I wonder about is, why it sort of gets back to the question of, the human, you know, the nature of being human and the code of life, the code that, we find in all human cells is mostly non-coding. We call it non-coding DNA, meaning that most of it doesn't encode proteins. And in fact, one of the surprises when the human genome was first sequenced was how few genes we find in the human genome. And yet there's an incredible, obviously, complexity to making a human being and all that we associate with that. And so one of the questions that I would love to answer, I don't know if I ever will, but I'd love to know what's essential in human DNA. What parts of it really are critical for the development of human being, and that'll be hard to answer. But I think one could certainly imagine doing experiments where you figure out what subset of DNA in a human cell is necessary to make a certain kind of cell, like a liver cell or a brain cell or a lung cell. And also, what are all the other molecules that you need besides proteins to do that? I love RNA, so I always want to know what RNA is doing in cells. So I think when I imagine what'll I do when I'm sort of of a certain age, whatever that age is, and have maybe I'm an emeritus professor and I have a little research operation going on. What would I do? And that's the kind of thing I'd like to go after, just sort of pie in the sky questions like that. Because I'm fundamentally fascinated by what makes us human. I think it's so interesting to think about that from a chemical sense, right? What's the chemistry of that, and how do we even ask that question in an experimental setting? I think that would be a great challenge. My parents have both died, sadly. So my father passed away in 1995, so he never knew about this. And my mother passed away a year ago, but she has been in a state where she didn't really know what was going on for a number of years. So she really did not ever know about CRISPR either. So sadly, neither of my parents knew about it. However, I kind of feel like, they knew me and they knew my passion for science. And I don't think at some level, they might be surprised or whatever about the details of all of this, but I don't think knowing me, that they would be surprised that I guess embraced the opportunity that came my way with this technology, right? That there was an opportunity to be involved in the discussion about responsible use, to be involved in doing podcasts like this, to help teach kids about science and why we do it and why it's fun and those sorts of things. So I don't think any of that would surprise them. It's the journey. Yeah, absolutely. I've always found that it's, and this is true with anything in life for me, whether it's I'm on a hike or I'm working in the lab, it's the journey that's the fun part, right? It's not necessarily getting to the answer or getting to the end of the trail. It's more the process of doing that's fun. It's the steps along the way and the things that you figure out that make it so interesting. Things that you couldn't expect, couldn't anticipate. It's the feeling that you're onto something. The feeling that it's probably the feeling that Sherlock Holmes had when he was following clues and trying to figure out a mystery. It's that kind of feeling that you're gathering the information to try to solve this mystery or answer this question that you desperately want to answer. Oh, hands down, I'm most proud of the people that I've trained and the people that have come from my work. I mean, from my lab. I think that when I look at these younger people who are now working in their own right, running research laboratories, doing great work, teaching their students, making new discoveries. That's what I'm proudest of. That sort of that legacy of science that stemmed from just, originally just me starting my, opening up my shop at Yale University back in 1994. I think about it occasionally. For me, I kind of, I sort of feel that when I look at my life, there's sort of my life before CRISPR and after, before and after, because there was a profound change that happened for me, right after the publication of our paper in 2012. And I could see it coming then, and looking back on it, I can see it even more retrospectively, of just sort of this huge wave of stuff that, came to me, whether it was everything, the science, the patent fight, the companies that got founded, and learning about all of that, learning how to talk about our science in a way that was more accessible to people who are outside of science. All of that, has sort of happened since then. I feel so grateful. I guess that's the thing I feel the most is gratitude, that I would be involved in this and be part of it and have these opportunities. I think of myself as a perpetual student, really. I'm a student. I'm always learning. And these are all areas where I've had just enormous opportunities to learn things that I would have never learned or been exposed to otherwise. So it's been amazing.